Couple of announcements before we get started. The first two have to do with the biology department, our wonderful biology department here at PSU. Couple of things on here. Let's see, where are we? Undergraduate studies. Hopefully, most of you know this website. Let's see if we can actually get it to open. That would be nice. Salmon-Schulmerich Scholarship. Those of you who are pre-medical students, there's a really nice scholarship that John and Marcia Salmon-Schulmerich have offered through the biology department for biology majors. The deadline for that is February 1st, so that is coming up. Second thing I wanted to announce as far as the biology department is concerned is our regular Thursday seminar this week, which is going to be about species distribution modeling via a large number of species. I really not quite sure what this is all about, but I think it has a lot to do with mathematical modeling of various different species, sequences, etc. Remember correctly, this is a new member of the mathematics department who will be talking about this particular aspect of things. And this is a nice reminder that mathematics and biology are really kind of inseparable these days. We've been talking more and more about the human genome. We'll continue to talk more about the human genome throughout today's lecture as well. Massively big data, and the only way to deal with big data is to have mathematical tools to be able to deal with that. And that's one of the reasons we have this first new hire in mathematics, which is really cool, and secondly, thinking about the ways to use math and thinking about biological questions. Third thing has nothing to do with the biology department. It has to do with the lecture recording. A number of you noticed that there was this really bizarre drop in terms of the sound at about 40-some minutes into the the lecture last time, and then the last 10 minutes dropped off completely. Think yourselves lucky I didn't put in what was actually in the recording software. It was really crazy, and anybody knows Camtasia, please come and talk to me, because it was a very bizarre error that they don't even seem to be able to understand what it's all about. But what that means is, again, there's missing about 11 minutes of last lecture. If anybody has a recording of that that's not mine, please send it to me and I'll try and dub that in so we can actually have all of that material together. Which brings me to the last point, which is what's happening a week from today? Midterm, exactly, in here, same time, same place as normal class. It will only cover material through Friday's lecture. Monday will be a review. And that review is basically for me to try and do a better job of explaining stuff I didn't do a good job of explaining the first time around, and for you to ask me even more questions. So speaking of which, are there any more questions right now before we get started with today's lecture? No. Okay. So let's, my apologies here. I think the first slide in the lecture notes actually said lecture 6, because I didn't get around to changing that. It's really lecture 7, if we're keeping count here. This is just a quick review. We're going to go over this again. I'm talking about DNA structure. But today, the things that we hadn't gotten around to talking about yet were really these things here in red, the alternative histones, and literally chromosome localization, and then a little bit about metaphase compaction of metaphase chromosomes. As I mentioned, this is really, really new data as of last week in terms of understanding what's going on with compaction of metaphase chromosomes. And there have been some really nice results as far as that's concerned so far. Last time we mostly talked about nucleosomes, which are the core histones, and the DNA being wrapped around those. That's what we understand the most about. We've got high-resolution structures. We know exactly where each of those 150-odd hydrogen bonds are, charge-charge interactions. What are most of the interactions in histones? Are they specific or nonspecific? Nonspecific. The only time you have a specific interaction with a specific sequence of DNA with a nucleosome is when? When you've got stretches of T's and A's about every 10 base pairs, because that's one turn of our helix. That allows more compaction to happen in the minor groove. And so it's a structural thing. It's not a sequence specific. So the DNA can make that structure. That is why you have those particular pieces there. We haven't talked too much about topology. We'll talk more about topology actually probably towards the end of the replication stuff, which we may or may not get to on Friday. Epigenetic inheritance we'll talk more about today. This has to do with those modifications to histone side chains. And what are the most common of those? Lysine. What kind of modifications on lysines? Acetylations and methylations, exactly. And then if you have methylation, particularly on K9 of histone H3, that will bind to other proteins which will help make heterochromatin. We'll talk more about heterochromatin today, but the basic idea of heterochromatin is it's really compacted. And if it's really compacted, that means that you can't get the other proteins associated which you need for gene expression. And then again, this modification. So any more questions on this? And if you ask any more questions on this, you can all take out your clickers because we'll have a clicker question. And try and actually get it to work this time as opposed to last time where it really didn't work. Okay, close. Here we go. Acetylation of lysine 9 of histone H3 in one nucleosome is likely to cause loosening of the DNA around the nucleosome, other proteins to bind the modified histone, acetylation of histone H3 in neighboring chromosomes, release of histone H1 from the DNA, or A, B, and C. And feel free to discuss this. Can't believe there are only 26 people here. Ten, 5, lots of people voted at the last minute. Okay. This looks like something I need to talk about a little bit more because we're kind of all over the place. Except for the fact if you add up A, B, and C together and put them on E, it would actually get almost 100%. So what does acetylation do to lysines? Changes the charge on the lysine. What's the normal charge on a lysine? Positively charged. Interacts with the negative part of the DNA which is wrapped around it. So it's definitely A. Other proteins binding to the modified histone. I just mentioned that, I thought. It's the other proteins, it's the histone code readers that are going to bind to that. And so if you've got A and B already that are right, E is automatically right. And so you can go back and check C later. Just advice as far as the exam is concerned. And it usually leads to acetylation of neighboring histones because that histone code reader also usually associates with a writer which sits next to it, which will then continue to modify all of these histones. And this leads to spreading of whatever that original change to the histones happens to be. So the correct answer is E. Exactly. Thank you. Okay, so finish up with the histones. Mentioned before, what do we have? We've got H2A, H2B, H3, and H4. You will probably see potentially even in some of the other notes the canonical histones. These are the histones that are in the canon, originally supposed to have been the Bible, but yeah, these are the standard histones that you see. When I talk about canonical, I'll mean standard for these kinds of things. And if you have any questions about that, again, please raise your hand, ask me, send me email about it. So the canonical histones are, again, H2A, H2B, H3, and H4. Those are not the only histones you find. We talked about histone H1 last time, but there are a number of other histones which you also find in cells, and these are often associated with very specific parts of the genome and parts of a particular chromosome. So a couple of these are here these are not all of them by any stretch of the imagination, they all have this histone fold, or a slightly modified form of the histone fold. What's the histone fold? You haven't had your coffee yet this morning? Was too wet on the ride in? At least for me, it was really wet. So the histone fold are 3 alpha helices that are going to be wrapping around each other in order to form the nucleosome. If you have an alternative histone, you want to put a new histone into there, you're going to have it be compatible with all the other ones. So these have to have histone folds in order to be able to associate with normal nucleosomes. So you're talking about alternative histones, getting a nucleosome, you've got to have this histone fold. Now, what's different about these guys is the different colors that we have here in the tails. And so tails are going to be interacting with what? Other nucleosomes and other proteins. And so that's the real key as far as this is concerned. So we're specifically going to look at this one, SNPA, which is an alternative histone H3. There are other ones that you find in areas that are always being transcribed. That's H33, and then H2A has a few other ones as well. But the main thing here is they've got histone folds, they can fit into the nucleosome, but then different tails so they can interact with other partners inside the cell. So the best understood of these, I think, is the SNPA, which is the centromere-specific nucleosome. We haven't talked too much about centromeres yet, and we're not going to for most of the rest of the class. But the centromeres are just the part of the chromosome which you have a specific structure that forms in order to allow chromosome segregation to take place in mitosis. Turns out it's not very sequence specific, but very structural specific. Yes?
On the previous slide, is there a significance to having the histone fold between 2 tails versus just on the end and only having 1 tail?
Okay, I guess the question is, sorry to paraphrase here, but is there sort of a reason, quote unquote, why you always have tails on both ends of the histone fold, front and back? On the alternatives. Turns out if we go back and look at on that slide right here, if you go back and look at that previous slide, all of the histones actually have tails on either side of that histone fold. So that seems to be a pretty typical kind of process. So it's sort of the central part of the protein. Those are going to vary in their lengths, the tails, both N-terminal and C-terminal, but all of the histones have these kinds of things, at least all eukaryotic histones. There's some really cool archaeohistones that we're not going to talk about that don't have tails, but we're not going to talk about them. Yeah, in terms of the alternative histones, they've got tails, but those tails are a little bit different. And so in the case of the centromere-specific histones, these then associate with the DNA, and it turns out in yeast, this is a specific sequence. In most other eukaryotic cells, it's non-specific sequence. It's more of a structure, lots of repeated DNA. We don't quite understand why that is. But in this region, you'll have nucleosomes that have this alternative histone. It's supposed to be shown here as this little green bit right there. Now this SNPA can now associate with all of the proteins that are important for this chromosome segregation. And this is microtubules, kinetochore, etc. But important here is it's just one of those nucleosome histones which has gotten exchanged to have this now specific alternative histone, which is important for a particular function. Those other alternative histones that we talked about on the last slide are basically the same, only they're going to be interacting with different proteins than with different functions. Because of this, it's actually going to be pretty stable. If you have one part of the chromosome that's got these kinds of histones associated with it, it's going to stay that way for the most part, as opposed to things that change a lot, such as these modifications that are happening on individual tails, like histone H3, etc. Those are going to be a lot more variable in terms of how they function. So we mentioned, again, heterochromatin being really compacted, euchromatin being less compacted. We'll see some examples a little bit later on today But just as a reminder, looking at heterochromatin, euchromatin, mostly due to modifications, covalent modifications that are happening to histones. The covalent modification that happens to a histone does not change the genetic code, but it can be inherited. And so this is what people call epigenetic inheritance, because it's a modification which is not a genetic modification, but it can be passed on from generation to generation. And epigenetics is a really big deal these days, partly because we didn't learn about it until relatively recently. But this is just a specific example of a much more general phenomenon. Again, it's a change that happens to your DNA, or for that matter, anything else, that can still be inherited from generation to generation, but it's not a change in the genetic material, the DNA. So here, the example of heterochromatin. What happens here, you start out with a chromosome up here at the top. You'll have heterochromatin on this left side. On the right side, you have euchromatin. What do you have to have in the middle here? Barriers, barrier proteins to stop any kind of spread of the heterochromatin or euchromatin, because those modifications are going to bind to histone reader complexes that will attach to histone writer complexes. There's going to be a way of stopping which of these are here. You have chromosome duplication. Chromosome duplication takes place. All of the new histones that get added after replication, those are going to be unmodified. They're freshly made by the ribosomes, newly translated. But if they're next to an already modified histone, what's going to happen? They're going to get modified. So just guilt by association, you've done replication, now you have these modifications that take place, you'll end up with the same distribution of heterochromatin and euchromatin in the daughter chromosomes as you had up here in the first place. So it's a state of the DNA, not a sequence of the DNA, which is being inherited here. And we'll talk, again, more and more about this as we move through the rest of the class. Another thing to mention here is that the modification itself, which is supposed to be that little green square, in and of itself, that's not enough to get this change in chromatin, particularly for heterochromatin. You have to have other proteins, like this, I guess, sort of horseshoe-ish like protein right here, to associate with that modified histone. And it's only that complex of proteins which leads to the compaction. So it's not just that change in the histone that has compaction. You have to have other proteins there as well, which is kind of like what we just talked about in our clicker question a second ago. So because of this and these kinds of modifications, that was just a little tiny piece, sort of a snippet of a particular chromosome. You've got lots of these areas in any particular chromosome which can be having this epigenetic modification that, again, gets transmitted from generation to generation. As we'll see later, actually, it even gets transmitted from organism to organism. So you can have epigenetic inheritance from mothers to daughters and mothers to granddaughters. Really amazing stuff that we're just beginning to understand here. But as far as the molecular level, these are really just modifications that lead to the same kind of modification and positive feedback loops. And so that's what all of these things are about here. Positive feedback loops that are giving you a particular chromatin structure. And because of that, again, after replication, you'll see the same thing over and over. And that becomes really useful when we talk about lamp brush chromosomes and polytene chromosomes in just a second here. So quick review so far. These not expressed genes are in heterochromatin. What do we see for expressed genes? Expressed genes are in these so-called loops, and we'll look at the loops here in just a second. The last thing about today's lecture is that these chromosomes actually occupy very specific regions in the nucleus, and this was a big surprise until very recently. People only thought that the chromosomes kind of floated around in the nucleus. There's a lot more structure in the nucleus than anybody ever really thought. And so we'll take a look at some of the data for that in just a second here. But first, loops. So how do we know about loops? We know about loops because of a few relatively rare examples, but it turns out that these rare examples are rather applicable in much more general cases. The nice thing about these rare examples is you can actually visualize them with a light microscope. Most of the time for human chromosomes, etc., you cannot do this. So here, lamp-brush chromosomes are amphibian oocyte chromosomes that are ridiculously long, and 4 copies always stay together, and they're always in the same state. Now, oocytes are rather specific cells. They have to produce lots and lots of proteins very, very quickly. So what you see in these lamp-brush chromosomes is what we have right here, a bunch of staining down here at the bottom. This is just staining for nucleic acid, with a few of these loops that are coming out from this backbone, you can call it here. Often people will call this the scaffold, which holds all of these DNAs together. And again, the nice thing about a lamp-brush chromosome is it's all stretched out. This is not true in human chromosomes. But just in terms of visualization, now if you look at any one of these particular loops that's sticking out here, it turns out that this is a region of the chromosome where you have very large amounts of messenger RNA production and then the proteins that are being produced from that. And again, this is oocytes where you're producing very large amounts of proteins, one of the reasons that you seem to be able to see this. So they are, even though it kind of looks like here, most of the DNA is present in these loop structures. If you think about the overall fluorescence here, most of that fluorescence is down here at the bottom. So most of your DNA is not in these loop structures. Most of it's compacted. And this is going to be true for most of the time. Even think about the human genome. How much of it is protein coding? Very, very small amounts of it. The rest of it is usually in this compacted state. Yes? Okay, feedback loops versus physical loops. Thank you for pointing that out. So basically, what am I talking about in terms of these loops? And it's confusing that I'm using the same term for the feedback loops and the loops of chromatin. So in this case, the loops for the active expression, this is a physical loop, what you can see here. That's your physical loop. The feedback loop is drawn as a circle, but it's just being drawn as a circle. That's an actual information transfer that's happening there.
And that's what you were talking about with 2 slides.
Right, and so that's what we're talking about when we talk about modifications that happen to histones, which lead to more modifications of histones. Now, in the case of the euchromatin formation, a positive feedback loop is going to be dissociating that DNA and allowing it to form a physical loop, which will then lead to gene expression. Hopefully that makes more sense now. And I haven't further confused people. It's fine. No worries. You're probably not the only one.
The feedback loop is different than the positive feedback loop.
Okay, so far we've only talked, okay, sorry, there was sort of repeat the question here. The feedback loop is different than the positive feedback loop. We will talk about negative feedback loops later. Yeah. So, but feedback loops are a general case. Positive feedback loops are what we've talked about so far, because my modification that's keeping that modification. The negative feedback loop we'll talk about later. Yeah, Devin. This guy. Yes.
So is that schematic, or is that showing us something structural?
Okay, so this is completely schematic. This is not showing us anything structural whatsoever. And again, complain to the textbook authors about this. Yeah, I'm sorry about that confusion, but thank you for bringing it up. Good? Okay, so these loops are where we have expression. And when I talk about expression, now, unfortunately, it's going to be one of these terms that I use, other molecular biologists use. It really means making messenger RNA that will become protein. So here it's transcription that's going to lead to translation that'll lead to production of a particular protein. We also know that this happens in other kinds of chromosomes. It's not just these lamp brush chromosomes. So the best examples of these are in a number of insects, not all insects, but a number of insects, and best known of these is Drosophila, some of the flies, where you have what are called polytene chromosomes. So instead of the 4 chromatids lined up like we saw in those lamp brush chromosomes, these are literally thousands of copies, identical or more or less identical copies of each of the chromosomes that are lined up completely next to each other. And you see these in salivary glands of fruit flies and, in fact, a lot of different flies. And it's a kind of a barcode that you can use to identify those individual organisms with. And one of our faculty members here, again, recently retired, Lester Newman, looked at these kinds of things. And he calls them a barcode for identifying the individual insect. So it's a lot like a karyotype. Only here we're talking about many, many copies of the genome all lined up right next to each other. So it doesn't have to be in metaphase. These are interphase. These are active chromosomes that are all lined up next to each other. And if you look at those, they have a specific banding pattern. Again, not unlike what you would see in a karyotype. And in a few places you have these regions where there's basically no bands to be seen whatsoever. If you now look more closely at those particular regions, it turns out that those have a very high concentration of proteins that you need for gene expression, like RNA polymerases, and we'll talk about all of the other transcription factors a little bit later on. So basically you see something here which doesn't have a band. And the interpretation of this is another region of the chromosome where you've gone from compacted to non-compacted. And in that non-compacted region, which you can't see under the microscope anymore, that's where you have all of the transcription going on. And so another indication that where you have active DNA is where it's looped out relative to the rest of your genome. So people also talk about these as puffs. You just see them as a region in these polytene chromosomes where not everything's lined up. It's no longer lined up anymore. Sorry. Yeah, so that's exactly right. We're looking at a couple of examples now on where we can see, and I mentioned this before. DNA is ridiculously inefficient. Watson and Crick came up with this wonderful model, which is great, except it's 10 base pairs per turn, and we've got 6.5 billion bases in each one of our cells. You've got to compact it, and most of the time it's really compacted, but occasionally you also have to be able to make a RNA from that particular gene. So these are just examples of talking about that. And we'll talk about a little bit more of the mechanism. We talk about gene expression again later on in the term. Okay, so last thing I want to talk about as far as these chromosome structures are concerned is even with this heterochromatin, and most of the time being compacted, again, you can see them nicely in frog oocytes, you can see them nicely in salivary glands in certain insects, but most of the time in a eukaryotic nucleus, say a human cell, mouse cell, etc., you can't see these things until you develop really neat techniques like we talked about with karyotypes, where you can now use a fluorescently stained piece of nucleotide sequence, which can hybridize with base pair to a specific sequence in each of the individual chromosomes. So that beautiful multicolored karyotype of all these different chromosomes, you can now do this also for interphase, not metaphase, and just look at a nucleus. And so this is a nucleus that has exactly those same stains that have been applied to it. And one of the things that's here, now the order of all of these things is completely irrelevant. It's going to be different in any different nucleus. But what's important here is that it's a rainbow. They're all separated from each other. So chromosome 1 doesn't overlap with where chromosome 2 is, and so on and so forth. So there are specific regions inside the nucleus. Again, it's going to vary from nucleus to nucleus, different cell type, etc., but they're all separated from each other. And again, this makes sense from the point in the back here. You're talking about releasing specific parts, expanding parts of the genome in order to get them to be transcribed, and the other ones being compressed. In order to have things compressed, you have to have ways of keeping them in particular places. And so the scaffolding, all the other protein that's present in the nucleus, is really helping with keep these particular chromosomes in individual locations. And there's some other techniques they go into the textbook about how you can look at where these particular chromosomes are relative to each other that we're not going to cover here. But the basic point here is that chromosomes are specifically localized in the nucleus even when they're in a non-expressed state. Yeah, your question.
Yeah, so actually you're saying this conservation or like separation happens even when the chromosomes are dissociated to be replicated before like division as well, or are they intermixed like mixed during that period of time?
Okay, maybe there's some confusion here, and probably because I didn't say things properly. So I'll try and explain it again and correct me again if it seems to be confusing. So the point here is that when we looked at the karyotypes before, that was metaphase chromosomes, so completely condensed. And in that case, yes, clearly they're going to be in specific locations. What people thought is before they undergo that last step of metaphase condensation, people thought they were all mixed together. That each of those extra pieces, these extra loops, were kind of mixed in with each other. That turns out not to be the case, and this is really rather surprising. And so that's what this is supposed to show. So you always have compaction. You've got to have compaction of your DNA, otherwise it's never going to fit in the nucleus. But it's not that extreme compaction that you get with the metaphase chromosomes. So basically, even when they're slightly compacted, which is maybe a better way of talking about it, they've still got this specific localization that's going on. Yeah?
Does this correlate back to structure and function? Is that they're in a specific region in the nucleus when they're in the metaphase because there's, say, a liver cell or a gut fragment or
Okay, so the basic question is there a sort of structure-function relationship relative to this? And this is a perfect lead-in to the next slide. Thank you. So I didn't pay her off here. So the basic thing here is, okay, so getting back to your compaction question. So compacted is, at this point, it's not actually being transcribed. What about transcription taking place? And so, yeah, look, they've got these nice, subnuclear localization going on. What happens to a gene when it starts to be expressed? So you've gone from that somewhat compacted to even less compacted, probably loop formation. If you look at this, this is where we have just looking at a single chromosome and now a single gene which is in it. So this is one copy of the chromosome right here, and that single gene is the little pink dot. If you look at now a nucleus where that gene is being expressed, it turns out that gene has now gotten moved to another part of the nucleus where it's now associating with the transcription proteins. So there does seem to be some subnuclear specialization, probably where all of those transcription proteins are. People talk about these as like a transcription factory. So those active genes, not only are they being made into loops by going into euchromatin, but somehow they're getting moved, and probably this is through just a diffusion process, but then getting captured by all of the active proteins right in the middle of the nucleus. But that's only a tiny piece of each of these individual chromosomes. So you're just doing the general staining, you're not going to see it. And in fact, here, if you look at it's maybe a little hard to see here, but these pink dots right here, when they're being actively expressed, are not where the rest of the chromosome is. So it's that little piece which has gotten moved, and it seems to be just diffusion floating around to this place in the middle of the nucleus where you're getting transcription taking place. So yes, there do seem to be regions of the nucleus that are important for different things. And we'll talk about replication too. It seems the same thing is happening there.
Well, it looks like the pink dot has migrated not kind of from the center of where it was, and it actually it's moved out. It's super cool.
Yeah, it is. I completely agree. It's super cool that that seems to have moved. Yes.
So when the DNA is in euchromatin, is it still in those 30 nanometer filaments? Is that kind of its default state when it's in interphase?
Yeah, so the question here really has to do is what's the default state of chromatin when it's in interphase and euchromatin versus heterochromatin? The answer to that is we really don't know. But for the most part, it seems to be the 30 nanometer filament. So something here in terms of most of the time, if it's not in a heterochromatin form, which most of the genome is in heterochromatin, so it's more compacted, but if it's in a euchromatin form, it's mostly going to be a 30 nanometer filament. To get it actually expressed, i.e. getting all of these extra proteins associated with it, it's probably becoming even more dissociated than that. But that's for those expressions. Right? In those loops, then it's going to be even less condensed, yes. Okay, so now we'll flip this over and talk about the very last bits of condensation. When you go from the euchromatin and heterochromatin that you have in a normal chromosome in interphase, how do you go to the metaphase chromosome? And again, I think I mentioned last week, we don't know very much about it, but now we know a lot more. So a couple of important proteins here, and mostly these are the so-called condensins. And what does it do? It condenses the DNA. So actually a semi-reasonable name for a change here. These particular proteins are called SMC proteins. That name is not important, but what is important is that these have very long coiled coil structures. Coiled coil is just another name for leucine zipper. Alpha helices wrapped around each other. Because every 7 amino acids, they've got a large hydrophobic. And what that means, they can have these long tails or hooks or whatever you really want to call them that also have ATP hydrolysis here in a separate domain of the protein. So one of these domains is this coiled coil, sort of the claw, and then a head domain here, which is important for ATP binding. What happens here is condensin grabs these individual loops of DNA and then associates with other condensins in order to pull everything together to get your final metaphase chromosome. And so that's shown in a little bit more detail here. There are actually two different condensins. Condensin 1, which will grab the loops of DNA, and condensin 2, which stacks all of those loops on top of each other. So if you look in the middle of a metaphase chromosome, the condensin is all here right down the middle of your chromosome, and the DNA, which is in sort of the purple or bluish here on the outside, is kind of on the outside. If you look here, the DNA are these relatively thin pieces here in an electron microscope, and condensin is present down here right at the base of each of these loops. And so this is the video that I mentioned. See if we can actually get it to play. In terms of the structure that was literally published last week in Science in terms of looking at some of these structures. So here is their model. Mystery for 136 years. Yes, I don't approve of this extra ad down here at the bottom. You break this down, look at those individual loops. Here's that individual loop. And the condensin, that's condensin 1, which is pulling that loop together. These guys all associate into a helix in the middle of one of these metaphase chromosomes. And then you have condensin. Those loops, again, condensin 1, different colors here, stacking on top of each other. Then we're going to see condensin 2, which is going to pull these loops together. That's the blue circles, which you can see on here. Those blue circles are pulling that loop in together. And that's where that ATP hydrolysis is important for. It's pulling the individual loops together. You pull those loops together, you line them all up on top of each other, condensin 1 in red, condensin 2 in blue, and then you end up with these compacted metaphase chromosomes. And again, the link is in the notes to take a look at this. It's really that interaction with the condensin 1 and condensin 2, which is really the big breakthrough that happened here in terms of this structure. Okay, we don't need this anymore. Bozeman science. I don't know about Bozeman science. Okay, so as a review, basically what we talked about last time, talked about today, start out with your DNA up here at the top, B-form DNA, the model made, from Watson and Crick's structure, all the way to the metaphase chromosome. Yes?
I was just a little confused on the condensin 1 and 2. So the condensin 1 grabs the loops of chromatin, and then condensin 2 grabs onto that loop and makes more?
And makes those loops smaller.
Okay.
So condensin 1 is making the loop in the first place. Condensin 2 is pulling it together.
Okay, so how, is there a typical size the condensin 1 will grab onto, or is it just?
So the question is, there a specific size that condensin 1 seems to bind onto versus, and then having condensin 2 compact that? The answer to that is I don't know. There probably is. And if I went back and looked at the paper, I could probably figure it out. But I don't know. I'll try and find it out for you. Yeah?
You may have answered this, but is there a specific location?
Oh, so the question is, there a specific location, and we can say specific sequence, that condensin 2 seems to associate with? The answer to that appears to be no. It's probably more of a distance thing. But again, the question is it a specific distance or a particular kind of structure? That I don't know, and I'll try and find out. Okay, so B-form DNA gets wrapped around the core histones to make nucleosomes. Again, we know a lot about this particular structure. Becomes a 30 nanometer filament. Exactly how this happens is not completely clear, but probably partly the nucleosomes stacking on each other. Certainly histone H1 plays a role, and certainly the tails of the individual histones are playing a role in this. This gets looped into, and sorry here, these are going to be different kinds of loops. Probably shouldn't use the loop term. This gets compacted into a piece of chromosome, and here's an extended form of the chromosome, and this is probably your 30 nanometer filament, still here wrapped together. Once that gets more condensed, this is where we're talking about the heterochromatin formation, that's going to be much more condensed, and finally, those get looped up through condensin 1 and condensin 2 into these metaphase chromosomes. So we've had a 10,000-fold compaction going from B-form DNA down here to your mitotic chromosome. But it's at this step where you have gene expression taking place. So this is a very dynamic process. We're going to have some of this at any given time where you have your gene expression. One other thing to mention down here, when you're at this really compacted state, no gene expression is taking place. So during mitosis, well, almost no gene expression is taking place. There are a couple of little tiny exceptions to that. There are always exceptions. This is biology. But for the most part, mitotic chromosomes are completely compacted and have no gene expression which is going on. Questions on this stuff, other than the ones we've had already? So now I get to ask you a question, right? Hey, the change in position of expressed genes in the nucleus is mostly thought to be due to diffusion, dissociation of histones from the DNA, movement to expression factories, formation of lamp-brush chromosomes, or formation of the nuclear pore, which we've talked so much about already. You're awfully quiet. Exam's not till next week. 10. Okay, we're split almost half and half, which I unfortunately realized is that actually I told you both things, right? So this is why we're confused. This is obviously not a terribly good question. And so what we'll do is everyone who had either A or C will get credit. Yay. So you can argue with your neighbors about this. I will try not to have questions like this on the exam. I do a lot better job of proofreading them for final exams when we do this. So I'm actually not going to give points at this point for this question. I'll do that later on offline. Pardon? So actually I was looking for C, but I said A. So again, given those issues, and I also realized that as I was lecturing. So, of course, I made these questions before I started talking to you. Okay, so I think we'll move on from here. Hopefully, we've covered all of this. Just the last little bit. Again, any more last-second questions? I'm not going to ask you any more about this. So, wanted to talk a little bit about the human genome, and this is again, a whole course in and of itself. Just as a reminder here, the important parts of the human genome, again, one can argue about what's important. I will tell you that it's this 8% of genes in the genome which are clearly like retroviruses, so viruses are most important for the human genome. But there's this 1.5% of actual protein coding sequences. These are the exon sequences, which are present here at the very end of the genome. This is not an end of a genome. They're spread out through the whole genome. This is just a cartoon looking at that. But one of the really surprising things when people did the human genome sequence, they were looking at the sequences in the human genome. Most of our genome is these repeated sequences. And up to here, including things like the centromeres, but lots of lines and sign elements. We'll talk about these later on when we talk about mutations. These are the transposons that are moving around in our genomes. In fact, they don't move around very much, probably partly because of all the heterochromatin stuff that's been really compacted. And there's this really interesting question that comes up here is, well, we just said, DNA is really inefficient. It's got to be compacted like mad. Well, why the heck do we still have all of this genome around? Why do we have all this extra junk DNA? Because of all these compaction and kinds of things. You would think evolutionarily there'd be a lot of pressure to get rid of this extra DNA. And interestingly enough, that doesn't seem to be the case. One of the things that is mentioned quite a lot, there's a whole section of the chapter that we're not going to cover in the class, talking about genome evolution. There's quite a lot of this non-repetitive DNA that we really don't know what it's doing. And a lot of people talk about this as the junk DNA. Now, here, these repeated sequences, they may or may not be junk. These non-repeated sequences are probably not junk, but we really don't know what they're doing. And so that's a big open area of research, trying to figure out what exactly is going on under these kind of situations. So that's DNA. Yes, wonderful. We know all about it. Answer lots of questions next week on the exam. Good. Okay, so the next problem that we'll talk about is how to make more DNA. So every time you have a cell that divides, it's got to make more of its DNA. So we'll talk about replication, the end of today's lecture, and then the lecture on Friday. And again, that's wherever we get to there will be the material that ends up being on the exam. So a couple of things about DNA. As I think I've mentioned probably ad nauseum now, DNA is really not a great molecule as far as genetic information is concerned. It's really long. We'll talk a lot more about how it's very susceptible to changes, chemical changes that happen to the DNA. If you're storing information in a chemical form, any kind of chemical change is going to change that information. So DNA is very susceptible to chemical change and therefore mutations that happen. So lots of mutations happen. Actually, probably far too many mutations are happening. And we'll talk about it. Actually, it turns out there's the silver lining to that is that if you didn't have mutations, you wouldn't have evolution. So probably do need to have some of that aspect. So the DNA does turn out to be a useful genetic material. Hopefully, the fact that DNA is replicated for the most part semi-conservatively is not news to anyone. We'll talk about the data for that in just a second. How it actually works, this has to do with the DNA polymerases, particularly in terms of how they put the right nucleotide in, or I should say the correct nucleotide, and also proofreading. This stuff we're probably going to just end up getting to either on Friday or potentially even after the midterm here. So let's talk about mutations. Mutations are going to be a genetic change which gets passed along through the replication process and then to the next generation. What's interesting is that it seems to be about 1 in a billion of the replication events that leads to a change which is actually maintained and passed along to the next generation. That's amazing. If you could make, when I type, how many times do I make a typo? It's definitely not 1 in a billion. So what that means, though, interestingly, is that if you think about the human haploid genome, how big is that? About 3 billion. What that means is every time your genome is replicated, there are 3 changes that get maintained. And there are a lot of times your genomes get replicated going from that fertilized egg all the way to you, and then all of the cells that are continually turning over, blood cells, immune cells, etc. So there's a certain mutation load which is happening all the time. There are always going to be these particular changes. And people have done some back-of-the-envelope calculations. They say the maximum number of essential proteins you could have in the human genome, given this mutation rate, is about 50,000 proteins. And considering that we've got about 20,000 proteins, that 50,000 is actually really not that far off as far as that number is concerned. So we may have a few more, but probably not that many. Most mutations are bad. Most kinds of changes that happen to the DNA, and we'll talk about the genetic code later. Genetic code is great because you can kind of ignore a lot of the individual mutations that are happening. But for the most part, what happens in any one of these given changes is that you'll have that particular change is eliminated because it's detrimental to that particular cell or that particular organism. However, you're going to have a few of these mutations, as I mentioned before, in order to have some kind of variation upon which selection can actually take place. So that's the story with mutations, and my connection here has gone down again, so I have to go back up on the stage, which is where I hate to be anyway. So let's look at replication. So when you have you're such a good typist that you only make 1 mistake every billion base pairs. So semi-conservative replication was actually really originally proposed by Watson and Crick. They said you pull the 2 strands apart, each of those strands get copied. And this is what's the line at the end of their paper. It has not escaped our notice that this is potentially important. One of the sort of the major understatements in the biological literature. So 2 strands get pulled apart, and each strand of the double helix gets copied. And so here, one of the things that not in the textbook, just make sure you've got 5' and 3' ends. You remember these are anti-parallel. That's what came from their structure. 5' end here, 3' end there, 3' end here, 5' end here. And then they get replicated, pulled apart, one from 3 to 5, the other one from 5 to 3. As we'll see, this is not actually how it works. But that was their idea. The 2 strands get pulled apart. Each of those strands ends up getting copied. So this is just shown in a cartoon form here. You have replication. Unfortunately, the colors are a little hard to see here. Come down here, your angle, see if it works any better. So we've got the red and orange. Again, horrible color scheme here. Those are the strands that you start with, after you have, sorry, orange to start with, then red strands are the first one which gets made, then you have green strands in the next one, then you have black strands in the next one. And so you go from in your first generation, this is our first generation, you end up with 2 new strands and 2 old strands. So old is orange here, new is red. Next round of replication, you still have this orange strand, just 2 of those strands. You still have your 2 red strands from this last time, and now you have 4 green strands. The next replication, again, you've got 4 green strands, 2 red strands, 2 orange strands, and now you have the all the, let's see, this would be 1, 2, 3, 4, 5, 6. We got 8 there, so 16 strands of new. But the thing is, with those strands, they're all wrapped around each other, right? So they're wrapped around each other. You can't really tell which one's new and which one's old. The only thing that you can tell is whether it's a old old, old new, or new old. Okay, huh? Does that make sense? New old, new old. So just go back through here again. This is now my color coding at the top here. So this thing keeps losing its connection. So at the very top, again, these are old, old strands. And so if you replicate, now you end up with new DNA, which is added here through those individual nucleotides. With those individual nucleotides, you'll now see that after that first step, you now have 2 hybrids, half old, half new. And then the next round, it's only after the second round of replication that you start to have 2 strands of DNA, which is all new, relative to what you had right up at the top here. So if there was some way that you could figure out what was old and new, which has also been called the most beautiful experiment in biology, that would be a way to see if this was what was actually going on. That Watson and Crick's prediction, it has not escaped our notice that, was true or not. Now, it turns out that there were a couple of other models for how people thought this replication could take place. And one of those was this so-called conservative replication. And conservative replication actually makes a whole bunch of sense. You have your double-stranded DNA that just gets copied instead of pulled apart and each of those individual strands gets copied. And the reason that makes sense is you don't have to be pulling these whole strands apart. And we talked about this when we talked about the DNA structure. Pulling the strands of DNA apart is actually kind of hard. So if you can keep them together, then it might actually turn out to be a pretty good way to replicate your DNA. On the other hand, there's another possibility called dispersive replication, but basically before I get there, you can actually tell, if you have a way of figuring out what new and old DNAs are, whether this model is correct or the semi-conservative model is correct. And that's because after one generation, you have 2 molecules now here which are both old and both new. So, and then in the next generation, you have 3 that are new and 1 which is all old. So the semi-conservative, you split them up and you end up with new and old. The last model, and it turns out that this is the model which is used for DNA repair that we'll talk about after the midterm, so forget about it for now. But the idea here is that bits and pieces of the genome are being replicated, and it's not all or nothing at the same time. And again, this makes a lot of sense because you've got to pull those whole strands apart. You've got to replicate the whole darn thing. If we're talking about the E. coli genome, it's a single genome, 5 million base pairs that you do all at once. And actually it is all at once, starting from one place, which seems kind of inefficient. But if you do individual pieces, that actually would potentially make a lot more sense. It could potentially be a lot more efficient way to do this. How do you tell whether this is the case or not? Again, you look at new and old. Again, if you can figure out how to do this. the new and old after the first generation here actually turned out to be half old and half new. This is actually a little confusing because half old and half new is exactly what semi-conservative replication predicts. So if this dispersive replication is actually what's going on, you're only going to know after the second generation, because this is now going to be 3/4 new and 1/4 old, as opposed to split up, relatively speaking, to each other. So how do you figure this out? This is a technique called differential ultracentrifugation, and basically this was invented by Matt Meselson, who is the Meselson of the Meselson and Stahl experiment, the most beautiful experiment in biology. Any of you know where Frank Stahl taught and studied? Well, not studied, but taught until recently? Eugene. Actually, just recently retired from the U of O. Came up and gave a talk a couple of years ago. Really smart, ridiculously left-wing guy. So neat that some of these people are still around. I just love this part about molecular biology, that you can actually meet some of these people. So what did they do? They figured out a way to separate old and new DNA from each other. And so how did they do that? They made heavy DNA, and heavy DNA being a denser DNA, just by growing E. coli with specific precursors for the individual nucleotides. And a lot of this also gets back to the experiments which were done by Chase and Hershey, where they showed that DNA was a genetic material by having these specific nucleotides. In the case of Hershey and Chase, they were radioactive nucleotides. Here, these are the non-radioactive but still different isotopic kinds of nucleotides. And Meselson and Stahl chose nitrogen. One of the professors that I taught for as a teaching assistant at Berkeley, Günther Stent, used P32. He told me all about how his experiment completely failed. Otherwise, he might have gotten the Nobel Prize for this. Actually, Meselson and Stahl didn't get the Nobel Prize for this, but they're still very famous as far as it's concerned. So what they did is they figured out, use nitrogen. And if you look at this model up here, which you can do after class, if you look at number of nitrogens versus number of phosphates, that will tell you why nitrogen was a really good idea. The other thing as far as nitrogen versus phosphate is concerned is that the difference in the atomic weight of phosphorus that is the standard phosphorus and the isotope of phosphorus, which you can find, the difference there is smaller percentage-wise than the difference in nitrogen from 14 to 50. So what they did, separated the DNA from the cells and then put them into this analytical ultracentrifuge and then just looked at the different densities of DNA that they got out at the end. So the great thing about this experiment, as far as I'm concerned, is that they could make predictions in terms of those three models that I just talked about, semi-conservative, conservative, and dispersive replication. They knew exactly what their data should look like before they did the experiment. So this is, I think, one of the reasons people call this the most beautiful experiment in biology, is you know what you're looking for before you look at the data. So semi-conservative, after your first generation, you should have hybrid, it's half and half, and then start to see just new. So half old, half new, and all new. With conservative, you're always going to have some old and increasing amounts of new. If you're dispersive, you're just going to get a percentage more and more of each of these different pieces. So they did this with analytical ultracentrifugation. This is the centrifuge rotor right here. They also just called this the big machine. Most importantly, however, are the data, which they got out. The most beautiful experiment in biology. You start out with heavy DNA right here, and then after one generation, you see one band. After 2 generations, or approximately 2 generations, you start to see 2 bands. And as you go through generation to generation, you get more and more of this new DNA, but you still have some of that partially old, partially new. So this is really pretty incontrovertible evidence that semi-conservative replication was what was happening, at least in the case of E. coli. And we're 3 seconds over. So we'll stop here. We'll have a clicker question to start next time. So think about this stuff.